So the mission of the Translational Immunology Section, it's not just one. There we have multiple missions. The main goal is to utilize the most state-of-the-art advanced technology to develop and perform immunological assays that are aimed at analyzing samples that are collected from our clinical investigator at NIAMS. We're also doing research involving study the effects of one of the class of drugs that's been recently came in the market to treat many autoimmune and inflammatory diseases, the JAK inhibitors, and we're focused on trying to understand their function on specific type of immune cells so that we can gain better tailored therapy for patients. So our research furthers the NIAMS mission from the point of view that we support clinical investigator as well as non-clinical investigator in better understanding autoimmune and inflammatory diseases in general, which what NIAMS investigator studies, and we make them available the technologies that we have, which, as I said, they are state-of-the-art technologies, so they can actually answer the question that they have, and therefore, better tailored therapies for patients that suffer from these diseases. Some of the leading technology that we utilize that we make available to NIAMS investigators are multicolor flow cytometry. We have new instruments that can measure up to 40 different parameters, so 40 different markers on immune cells, for example. We have a way also to do some transcriptomic analysis, which means measuring the expression of specific gene by looking at their messenger RNA expression in specific tissue or cell types through the nanostring encounter system. But we've been very fortunate to collaborate not only with people in NIAMS but also throughout the NIH. We've been involved with the discoveries and the characterization of many new diseases, most recently the VEXAS syndrome. Also, going back to the JAK inhibitors that I mentioned before, we did this study, a preclinical study showing that one of the JAK inhibitors that now is commercially available could have potentially be used to treat lupus, which is a very serious autoimmune disease. And actually our research in a preclinical model was then translated in a clinical trial that was performed by investigators at NIAMS. The most exciting thing about this field, of course, is that every year there's something new coming out, some new techniques, new instruments, new tools that we can actually apply to do the studies. And we're also integrating a lot our efforts with other groups at NIAMS, the light imaging section, for example, or the high throughput sequencing section. And by integrating our assay with their assay, we were able to do things that we were never been able to do before.